Chromosomes reading. Your body is composed of approximately 100 trillion cells. The intricate organism that you are today is the result of millions of cell divisions. Each cell division is a carefully orchestrated performance carried out by many kinds of structures. In this section, these structures, the chromosome, and how genetic information is transmitted from one generation to the next will be discussed. Chromosome structure. DNA, deoxyribonucleic acid, is a long, thin molecule that contains the information needed to direct a cell's activities and to determine a cell's characteristics. The information encoded in DNA is organized into genes. A gene is a segment of DNA that transmits information for a particular trait, like brown hair. A single molecule of DNA has thousands of genes, which are lined up like the railroad cars of a train. When a cell prepares to divide, the DNA molecule duplicates, coils up, and twists into a dense structure called a chromosome. If DNA did not coil and form a chromosome, a single DNA strand would be about 5 centimeters, approximately 2 inches long. This is too long to fit inside a cell. When the DNA coils into a chromosome, it becomes visible through a microscope. The chromosome looks like an X. The 2 copies of the DNA are called chromatids. Each chromatid contains the exact same genetic information. The 2 chromatids are attached near the center of the chromosome. This attachment point is called a centromere, shown in the diagram. The shorter end of the chromosome is called the petite or P-arm. The other, longer arm is called the Q-arm. It's called the Q-arm because Q comes after P. Human cells have 46 chromosomes. They come in 23 pairs. The 2 copies of each chromosome are called homologous chromosomes, or homologs. Homologous chromosomes are similar in shape and size and have similar genetic information. You received 1 homolog from each chromosome from your mother and the other homolog from your father. When a cell contains 2 homologs, each chromosome is termed diploid. Biologists use the symbol 2N to represent the diploid number of chromosomes in a cell. For humans, 2N equals 46. Not all cells are diploid. In the life cycle of animals, including humans, gametes, egg cells, and sperm cells are haploid. A haploid cell contains only 1 copy of each chromosome. The fusion of 2 haploid gametes forms a diploid zygote. A zygote is a fertilized egg, the first cell of a new individual. Being haploid ensures that when an egg and sperm fuse, the resulting zygote will contain the characteristic diploid number of chromosomes for that organism. Biologists use the symbol N to represent the haploid number of chromosomes. For humans, N equals 23. The table below lists the characteristic chromosome number for a variety of organisms. Please refer to your reading for the table. Chromosomes affect development. Each of the 46 chromosomes has thousands of genes that play important roles in determining how a person's body develops and functions. All these genes must be present in an individual's cells for the same reason that a car must have an engine, a transmission, and wheels to function properly. Therefore, a person must have the characteristic number of chromosomes in his or her cells. In most cases, humans who are missing even 1 chromosome do not survive embryonic development. The condition in which a diploid cell is missing a chromosome is called monosomy. And just as a car will not function correctly With 2 engines stuffed under the hood, a human embryo will not develop properly with more than 2 copies of most chromosomes. The condition in which a diploid cell has an extra chromosome is called trisomy. Deviations in chromosome number can be detected by analyzing a karyotype, the collection of chromosomes found in an individual's cells. A common chromosome abnormality is having an extra copy of chromosome 21. The traits produced by having an extra copy of chromosome 21 were first described in 1866 by British physician J. Langdon Down and are collectively called Down syndrome or trisomy 21 syndrome. The features that characterize Down syndrome include a short stature, a round face with upper eyelids that cover the inner corners of the eyes, and most significantly, varying degrees of mental retardation. Down syndrome occurs in all racial groups with the same frequency, approximately 1 in 1,000 children. It is much more common in children of older mothers. In mothers younger than 30 years old, the incidence is only about 1 in 1,500 births, while in mothers 30 to 35 years old, the incidence doubles to 1 in 750 births. In mothers older than 45, the risk is as high as 1 in 16 births. The reason that more babies with Down syndrome are born to older mothers is that all the eggs a female will ever produce are present in her ovaries when she is born. As the female ages, the eggs can accumulate an increasing amount of damage. Males, in contrast, produce new sperm throughout adult life. What events cause an individual to have an extra copy of a chromosome? When a cell divides normally, each chromosome and its homologue separate, an event called disjunction. When normal disjunction does not occur, 1 or more chromosomes may fail to separate properly. This accident in chromosome separation is called non-disjunction, which results in one new cell receiving both chromosomes and the other new cell receiving none. Trisomics arise as a result of non-disjunction. In the case of Down syndrome, non-disjunction occurs with chromosome 21. Chromosomes determine your sex. Of the 23 pairs of chromosomes in human cells, 22 pairs are the same in males and females. These chromosomes are called autosomes. The chromosomes that differ between males and females are called the sex chromosomes because they carry the genes that determine an individual's sex. Sex chromosomes exist in either 2 forms, as an X chromosome or as a shorter Y chromosome. In humans and many other organisms, the genes that cause a fertilized egg to develop into a male are located on the Y chromosome. Thus, any individual with a Y chromosome is a male, and any individual without a Y chromosome is a female. In these cases, females are designated XX because they have 2 X chromosomes, and males are designated XY because they have 1 X chromosome and 1 Y chromosome. Because a female can donate only an X chromosome to her offspring, the male determines the sex of an offspring who can donate either an X or a Y. In some insects, such as grasshoppers, there is no Y chromosome. In such cases, the females are characterized as XX and the males as XO. The O indicates the absence of a chromosome. In birds, moths, and butterflies, the male has 2 X chromosomes and the female only 1. Alterations in chromosome structure. Although very rare, changes in an organism's chromosome structure do occur. Some alterations cause mutations, changes in an organism's genetic material. Some of these mutations are illustrated below. When a fragment of a chromosome breaks off, it can be lost when the cell divides, causing a mutation called a deletion. As a result of deletion, a new cell will lack a certain set of genes. In a mutation called a duplication, the chromosome fragment attaches to its homologous chromosome, which will then carry 2 copies of a certain set of genes. Sometimes the fragment reattaches to the original chromosome in the reverse orientation, producing a mutation called an inversion. Or the fragment may join a non-homologous chromosome in an event called translocation. References for this reading are from Curtis and Barnes, Invitation to Biology, published by Worth Publishers, Incorporated in 1981. Johnson and Raven, Biology, Principles and Explorations, published by Holt, Reinhardt, and Winston in 1996. Tolley, Modern Biology, published by Holt, Reinhardt, and Winston in 1989.